Baking bread essentially just involves the mixing of 4 ingredients. However, along with that, there is a surprising amount of chemistry. The basic 4 steps to making bread are mixing the ingredients, consisting of flour, water, yeast, and salt, to make dough, kneading that dough, letting it rise, then baking it. On the surface, this process seems simple and easy, but on a molecular level, it is actually very complex.
When it comes to mixing the dough, each ingredient plays a more important role in the taste of the bread as well as the chemistry in the process. Flour is 10 to 15% proteins, including the class of proteins called glutenins and gliadins, mainly built of amino acids. They are also referred to more commonly as gluten. When water is added to these proteins, they are able to line up with each other and interact. They form hydrogen bonds and disulfide cross-links between their chains, forming a gluten network within the dough. The salt in the compound ascorbic acid, or vitamin C, helps strengthen the gluten network and makes the dough more elastic.
The gluten network is important when it comes to the rising of bread, but the rising won't occur at all without the yeast. Yeast contains enzymes that break down the starch within the flour into sugars. Yeast first uses amylase to break down the starch into maltose, and then uses that maltose to break down into glucose. The glucose essentially feeds the yeast so it metabolites and produces carbon dioxide and ethanol.
The excess sugar produced by the process also participates in the Maillard reaction. The Maillard reaction is a series of reactions between sugars and amino acids that happen once the temperature reaches 140 degrees Celsius. The products created by this add flavor and help to form the brown crust of the bread. Focusing back on the rest of the sugar, the sugar metabolism, though, the ethanol produced is the same as the alcohol found in alcoholic drinks, although it is expelled from the dough during the baking process, so you can, in fact, not get drunk off of bread. The carbon dioxide is also diffused through the dough and enlarges pre-existing air bubbles, helping the bread to rise.
The gluten network is involved in the trapping of the carbon dioxide, but the proteins and the lipids in the dough are involved as well. The process, of course, is different for sourdough breads. Sourdoughs involve starting with a mixture of flour and water. After the initial mixture, the natural microbes in the flour begin to grow, and the mixture is regularly fed with more flour and water, eventually ending with a mix of bacteria and yeasts. The yeast in sourdough is different than baker's yeast used in normal bread, as they are more acidic tolerant due to the acidic compounds produced by the bacteria. These yeasts are unable to create maltose, but the bacteria is able to produce food for both the bacteria and the yeasts. The ultimate results are the same, although the taste is altered by the bacterial metabolites.